All right, so last class we left off talking about the glycosylation that can occur on different peptides that are being have been translated, and how those glycosylation events oftentimes occur across the ER as well as the Golgi. In case there was any confusion, I think perhaps on my part, there's an effort to try and get through these slides quickly because there was so much to cover across these two lectures. I wanted to take a step back or rewind 2 slides to make sure that the point of the last 2 slides we discussed last class is clear. The slide entitled "Who Cares About Glycosylation?" Seemingly, it sounds like there's small sugar molecules that are added onto proteins as they're translated. What does that ultimately matter? This slide where I discussed that glycosylation was able to sequester or hide the binding epitope for these antibodies was meant to just try and demonstrate that these glycosylation events are able to regulate and in some ways impact the function of a protein, as well as specifically the sites that are really important for the function of that protein. We talked about in the previous lecture how O-linked glycosylation oftentimes occurs on serines and threonines. If you think back to the second lecture this semester, which might feel like a year ago now, we talked about how serines and threonines are really important amino acid residues that are phosphorylated. And so this is an example where perhaps for this protein, or for proteins that have O-linked glycosylation events, that if there's sufficient glycosylation events across their serines and threonines, that perhaps kinases that want to phosphorylate there might not be able to. And to further demonstrate or show to you the importance of glycosylation events, I tried to walk through and describe our different blood types and how they are determined, and how there's this native. Let's see if I can. Sorry about that. This native lipid that's on the surface of our red blood cells that has this sugar residue on there, and that the same gene that encodes for an enzyme has 3 different alleles or 3 different versions, where the O allele has a frame shift mutation or a frame shift change in that gene that elicits an inactive gene or inactive enzyme. And for people with the O allele or O blood type, this is what that sugar residue on the lipid on the membrane of our red blood cells looks like. If you have the A allele, you have the GalNAc-transferase and N-acetyl galactose that is added on to the sugar residue for the A allele, and for the B allele, you have a galactose transferase that adds a galactose there. So these are sugar modifications or glycosylation events that can occur that regulate our blood types. So we talked about co-translational import, and we talked about post-translational import. Again, co-translational import describes how, as a peptide is being translated, while it's happening co-translationally, it's being imported into the lumen of the ER, and post-translational import is happening out in the cytoplasm with free ribosomes that then will be directed elsewhere. The few examples of post-translational import are when things that lack any signal sequence or any kind of sequence are just going to be translated in the cytoplasm on a free ribosome and just left and produced and left in the cytoplasm. Or they might have that nucleotide or mitochondrial sequence. The 3 examples of co-translational import, we talked about secreted peptides, peptides that have a transmembrane region that is the stop transfer that encodes for it to be moved into the membrane of the ER. And the final one are those that are sent over to the lysosome that encode for lysosomal enzymes. And the way that is part of the way that's determined, in addition to the signal sequence that prompts co-translational import, there's these lysosomal enzymes, or also referred to as lysosomal hydrolases. They mediate hydrolysis to degrade whatever is in the lysosome. What ends up happening here is that they are, through a series of enzymes, they are tagged with a sugar that's called mannose-6-phosphate. And the way this happens, these other sequences that we talked about, the signal sequence or the stop transfer, those are signals in the primary sequence, the linear sequence of the amino acid that's recognized. Here, there's not a signal at the primary structure level, for lack of a better phrase, but rather in the tertiary structural level, where there's something called a signal patch, this physical signal that's detected that signals for mannose-6-phosphate to be added onto this lysosomal hydrolase. And what ends up happening is that within the Golgi, there's a receptor for mannose-6-phosphate, the mannose-6-phosphate receptor. And once this hydrolase that has mannose-6-phosphate on it binds to the receptor, that'll prompt the preparation for a vesicle to form off of the Golgi. Now, we talked about before, during endocytosis, we talked about clathrin-coated pits that help with the formation, the invagination, and the bringing in of receptors with a ligand bound into them. Here, rather than clathrin, something called codomer. So there's a codomer-coated pit that begins to form on the Golgi, where the lysosomal hydrolase with its mannose-6-phosphate binds to the receptor, and it'll create a vesicle that then will fuse with an early endosome, and then will eventually mature to form a lysosome. And so that's how these lysosomal hydrolases, as they're being translated within the ER, will then get modified based on the signal from the signal patch for mannose-6-phosphate to be docked on. It'll bind to a mannose-6-phosphate receptor to prompt the formation of a codomer-coated pit to release this vesicle that fuses with an early endosome to promote the formation of a lysosome that has this lysosomal hydrolase. And so moving on to some of the other examples of post-translational import here. A couple of the other examples are nuclear localization that occurs, as well as mitochondrial targeting. We're going to focus on nuclear localization, and then we're going to try and tackle Noonan syndrome to wrap up the lecture. So there are oftentimes proteins that are being trafficked in and out of the nucleus. And if you can imagine, this process is pretty important because our nucleus possesses are really important and really important DNA that encodes for a lot of our genes, that should something get in that's not meant to be there, that could be problematic. And so you can imagine that there's an evolutionary advantage to having tightly regulated processes that regulate and traffic this transport. So we're first going to talk about the import of things into the nucleus and the exportation. So nuclear proteins that are going to be trafficked into the nucleus contain the nuclear localization signal. And what's going to end up happening is that this process, everything that is going to be occurring across this transport is going to be regulated by concentration gradients. And so when we have proteins that have been translated by free ribosomes, this was not by ER-bound ribosomes, but rather free ribosomes. Once that happens, this protein with a nuclear localization signal is going to be in the cytoplasm, and will want to, its fate is to be in the nucleus where it has its designated function. But what's important to know is that these proteins that are being synthesized that have this NLS signal, a lot of them are in the nucleus. So it'd be moving against the concentration, it'd be trying to move up a concentration gradient, which it cannot do. So it needs, for lack of a better phrase, it needs a dance partner. It needs someone to help carry it across into the nucleus. And so the way that happens is this is going to happen via a protein called important. Important now has a concentration gradient that will move into the nucleus and recognizes the NLS. So important here recognizes the NLS, the nuclear localization signal, will bind to it. And now this NLS possessing protein, which serves as a cargo, will move with important down its concentration gradient through, this is called this NPC is called the nuclear pore complex. It's this, it's these really large macromolecular protein that helps regulate what moves in and out into the nucleus. And so it'll important with its cargo protein that has the NLS will ride down the concentration gradient into the nucleus now across the NPC. Now, what's important is that important is going to bind to a G protein called RAN, for which it has a higher affinity. When RAN is loaded with GTP, that is what important has a higher affinity for. So important to continue the analogy is going to leave its dance partner for a better dance partner. And so it's sad, I know, I'm sorry. So important will then bind to RAN, GTP loaded RAN. Now the concentration gradient for GTP loaded RAN with important is going to ride down its concentration gradient through the NPC back into the cytoplasm. Here is where the GTP will be hydrolyzed. And once that hydrolysis occurs, important will disassociate. Its affinity was for GTP loaded RAN. Once that GTP is hydrolyzed, it'll leave and look for a better dance partner again. And so this entire process is how NLS possessing proteins that have just been translated will find a way into the nucleus. They'll ride with down importance concentration gradient into the nucleus. Now, for things leaving the nucleus, it's pretty much the same, but the process is in reverse. And so there are proteins that are going to possess a nuclear export signal. Now, what's important to know is that there are going to be proteins that might possess only one of them, but oftentimes they will likely possess both in an effort to provide tight regulation for proteins that are going in and out of the nucleus for their designated function for each of those corresponding proteins. And so what's going to end up happening now is that for a protein, and in addition to that, I should say, I mentioned in passing last class that proteins that are below 40 kilodaltons can passively diffuse into the nucleus. And so this export process is how we also can remove those proteins. Because again, we want to, it is evolutionarily advantageous to have a process that tightly regulates what is allowed and not allowed in the nucleus. Because again, that's a really important space in each of our cells. And so each of them will have a nuclear export signal. And what will end up happening is it will bind, this protein with the NES will bind to GTP loaded RAN, but this time with a protein called exportin. So when importing, it was important. With export, it's exportin. So the NES possessing protein will bind to exportin that's within GTP loaded RAN, ride down the concentration gradient. Once that GTP is hydrolyzed, exportin will dissociate from RAN, and now that protein will also dissociate from exportin. Exportin will ride down its concentration gradient back into the nucleus, ready to repeat this process. So this process is pretty similar, just in reverse, and is dictated by concentration gradients. Are there any questions on the import or exporting of newly translated proteins across the nucleus? Yes. That's exactly right. They're going to be amino acid sequences that are recognized by importans or exportins. Cool. The concentration gradient is just the amount of the concentration or the amount of a certain protein within a compartment. And so oftentimes it will want to go from its high concentration area to a lower concentration area. It's hard for it, it won't want to freely diffuse to where there's already a high concentration of it. And so to address that, there are these proteins that help shuttle and ride their own concentration gradients to traffic these proteins that have been just translated, or to export proteins that have just passively diffused in that shouldn't be there. Any other questions? All right. This is a bit of a graphic to help illustrate what the nuclear pore complex looks like. It is a 50, not kilodalton, 50 megadalton assembly. It is incredibly large. It has 30 distinct proteins that we call nucleoporins or NUPs, and across eukaryotes and prokaryotes, there are about a few dozen different species, but each nuclear pore complex possesses approximately 450 individual proteins, and so it's a really large, really important macromolecular complex that regulates and actually has feelers kind of like in the pore, within the lumen of the pore, that helps regulate large proteins from entering in. 40 kilodalton smaller proteins are able to passively enter through, but it helps ensure that only proteins that are meant to be there go through, and if smaller ones get in, we use the export process to traffic them out. So for newly translated proteins that are meant to be targeted for the membrane, yet don't have a membrane spanning domain, are oftentimes undergo a post-translational modification to help localize them and tether them physically to the plasma membrane. One of those key modifications is called prenylation. Prenylation, typically occurs on the C-terminus of the newly translated protein. And it's the addition of this hydrocarbon tail, and it typically occurs whenever there's a signal on the C-terminus of a CAX box. So CAX is cysteine, alanine, and then any amino acid. That's typically what X means. And so any newly translated protein that has a CAX box on its C-terminus is going to be prenylated, and that modification is going to tether it to the inner leaflet of the plasma membrane. Remember, it's a phospholipid bilayer, an outer leaflet that faces towards the outside, and an inner leaflet that's on the internal side. This will tether it to the internal portion of the plasma membrane. There are a few different species or a few different kinds of prenylation events, and each one is determined by how many prenyl groups there are. And so you can, as a way to kind of help determine how many prenyl groups there are, you can kind of look for the, it'll end up being a methyl group on top here, but you can look for basically the double, it'll be a single bond, double bond, and then a kink as a result of that double bond with the methyl group up here, and you can basically count each of those as a prenyl group. So journalation are two prenyl groups, two prenyl group modification. Farnesylation is a 3 prenyl group modification, and then whenever there's 4, they couldn't come up with a name for 4, so they did journalation modification. And so these are going to be the number of prenylation events that occur on the C-terminus in response to the presence of a CAX box. Another modification is the Glycophosphatidyl inositol anchor. This is also called GPI. We talked about GPI anchored proteins being present within these lipid rafts and these cholesterol-rich domains. What again, almost feels like a year ago. In the earlier phase, much earlier in the semester, when we talked about the cell membrane. And these GPI anchored proteins, the GPI anchor tethers it to the outer leaflet of the membrane. So it faces, and it's on the C-terminus. So that means the C-terminus is anchored to the outer leaflet, and this protein is facing out towards the cell. There's a protein that's near and dear to my heart that my entire postdoctoral research has been focused on. That's a GPI anchored protein. And so these are typically not only facing outward and anchored in the membrane, but in these lipid raft domains that are sites that have very active cell signaling that's occurring. The last that's going to be of interest to us as we try and pivot back towards Noonan syndrome is called meristylation. And typically it's, instead of on the C-terminus, it is a modification that happens on the N-terminus that includes the addition of this long hydrocarbon tail wherever there's, again, we talked about how newly translated proteins, peptides, have methionine as their first amino acid. So whenever there's a methionine and then a glycine, there's the possibility for myristoylation to occur on the N-terminus. And that tethers it to the inner leaflet. We talked about RAS and how that's an important monomeric G protein and the RAS-MAPK signaling cascade. And RAS is farnesylated, which tethers it to the cell membrane on the inner leaflet. And what we're in, and we're going to talk about how that can go wrong during Noonan syndrome. So again, prenylation is on the C-terminus. And so there we go. On the C-terminus end, the farnesylation or the journalation of this peptide is going to create this hydrocarbon tail that's wedged into the inner leaflet here and localizes it on the plasma membrane. And what's going to end up happening is we talked about how it's targets the CAX box, cysteine, alanine, and then some amino acid. The cysteine side chain is going to have, it's going to be what's important and what's going to be targeted during prenylation. And it's going to have this free thiol on the side chain there that's going to be targeted, and through this free thiol group, there's going to be a thiol ether bond that's catalyzed with the prenyl group, and it forms a stable covalent linkage into and wedges it into the plasma membrane. So myristoylation is typically an irreversible addition of a myristoyl group, and it's going to be via an amide bond because, again, it's in the N-terminus or the amine terminal end of this protein. It's, as you can imagine, it's catalyzed by N myristoyl transferase, and so on the amine group of the N-terminus of this polypeptide chain or this peptide chain, this myristoyl group is going to be wedged into the plasma membrane there. And what's really important to note is that it's that myristoylation that's going to be really critical in Noonan syndrome. So taking a step back and thinking back to the RAS-MAPK signaling cascade, we talked about how RAS will activate RAF. RAS is a G protein that, again, G proteins are localized to the plasma membrane. I mentioned how it's farnesylated, so it's restricted to the plasma membrane. RAS and RAF will interact with each other, but what ends up happening is that there's also a scaffold protein that helps to aid in that interaction and help it occur a little bit more frequently. Scaffold proteins are a class of proteins where they will bind two separate proteins and help bring them together and mediate a chemical interaction. So I mentioned in the previous lecture how adapter proteins interact with two different proteins and bring them together. With adapter proteins, they don't mediate a chemical interaction, but scaffold proteins do. And so here, the scaffold protein SHOC2 will help aid in their interaction between RAS and RAF. They're not always doing it, but when they are present, they help aid in that interaction. They're not necessary, but help promote a more efficient interaction to activate RAF by RAS. Now, what ends up happening in Noonan syndrome is that SHOC2 has a mutation. We talked about kind of like the mutation nomenclature. Here, it's a serine to glycine mutation, and the serine is the second amino acid to a glycine. So again, the first amino acid is always a methionine. In this case, this serine is changed to a glycine. So we have methionine and glycine, which is usually what's targeted for myristoylation. And so SHOC2 is myristoylated. And what ends up happening is that SHOC2 now is restricted now to the plasma membrane because of this myristoylation. And it's going to help mediate a lot more activation of RAF by RAS. It's going to be bringing in RAF a lot more frequently to RAS. And it's going to be, the term we use is abherent activation. Abherent is kind of another fancy word for like dysfunctional. And so it's going to lead to overactive RAS-MAPK signaling, which lies at the heart of Noonan syndrome. If you have any further interest in the subject, Two people that are focused on protein trafficking are within BME, Dr. Jeff Saucerman and Dr. David Castle. Before I move on to our glycolysis lecture, I wanted to hit pause and say, are there any questions here? So Liam's hand go up and then I'll go to you next. Liam?
So what is the mutation on the shock? Is that it's isolated or that it changes?
It's a change from the serine to glycine. And that change exposes it to the modification of installation. Does that make sense? Great. Anna?
What exactly is the change that occurs with Metabolism? So I know it targets like a mixed by like pattern acid sequence, but what does it do to the amino acid chain?
So what it does is it's changing the hydrophobic nature of that protein, where now that that long hydrocarbon tail, I'll go back one slide, myristoylation is now covalently linking myristic acid to our peptide. And that's now going to create a favorable hydrophobic interaction to wedge it into the plasma membrane. And so in the case of SHOC2 is not typically a membrane tethered protein. It's typically within the cytoplasm and helping to mediate RAS-RAF interactions. When it's myristoylated, it's now wedged into the membrane. And now there's going to be, in some cases, in some myristoylated proteins, there might be a minimal effect, but in this case, it creates an overactive cascade where it's constantly bringing in RAF to bind RAS.
Okay, so it adds this myristic acid to it, and that confers like an element of hydrophobicity to it, which allows it to wedge it.
Yes. Are there any other questions? These are great questions. All right, I'll let you all catch your breath really quickly while I pivot to the next presentation. All right. Sorry that I didn't give you all a longer break. My hope is to try and get through as much of this as possible, just so that we can have finished that last lecture and finish today's lecture, so that we have sufficient time for the next subject we're going to talk about. So we're now going to pivot away from what we've been talking about. We spent a lot of time talking about central dogma, the extra dogma topics that elaborate on that, and protein trafficking to describe how these newly synthesized or newly translated proteins are trafficked throughout the cell. We are going to pivot now towards metabolism. And there's a lot of different elements of metabolism. And so we're going to try and tackle each large umbrella that falls under metabolism and talk about it within the context of obesity, different diets, as well as alcohol metabolism. So for obesity, as I'm sure many of you know, obesity is described as excess body weight. And it's an important public health problem because with how diets have changed and with how lifestyle has changed over time, particularly in the Western world, but now Western habits are expanding throughout the world, obesity is becoming the public health crisis, one of the leading public health crises internationally. Along with obesity, there's associated type 2 diabetes. We're now all intricately familiar with that after our transcriptional regulation lecture. Hypertension, which is high blood pressure. Stroke, which is blood clot in the brain. Hyperlipidemia, is elevated circulating lipid levels, and that's associated with atherosclerosis, which is the formation of a plaque within our bloodstream, as well as the downstream effect of that, which is a plaque in the coronary artery that leads to heart attack or myocardial infarction. And there's also correlation of some cancers with obesity. This is one point that we're about to tackle a few things that I wanted an opportunity to update. I think the next couple of minutes might be a little antiquated. An obese patient is described as being 20% more heavier than their ideal weight. And that's determined by their BMI. I'm sure many of you all can appreciate this, but BMI is defined by weight as it relates to height, in particular for lack of a better phrase, surface area. It relates to your weight to surface area. And that's not the best indicator of health or what your ideal weight should be. As I'm sure you know, many people have varying levels of muscle mass, and that is not the best indicator of what typically ideal weight should be. Rather than focusing on an ideal weight, I want to say all this, that I'm not a clinician, so like take what I'm saying with a grain of salt. But a lot of research and a lot of science is indicating that rather than focusing on BMI, focusing on body fat percentage, as well as the content or type of body fat that's present, is really important. And so that's a better indication of, for lack of a better phrase, health, because our adipose tissue and where those adipose depots are present, whether it's subcutaneously or around our visceral organs, is a better indicator of health, because that's going to indicate whether that adipose tissue is metabolically dysfunctional, whether there's a predisposition towards cancer or predisposition towards overall metabolic syndrome. So like there's kind of all of the things that we described that are associated with obesity, typically before they manifest is kind of under this catch-all phrase of metabolic syndrome. And so what's a better correlate for metabolic syndrome rather than BMI, while there is a strong correlation there, is on body fat percentage. So I'll end my soapbox rant there. But again, BMI is typically defined by your weight and height, or weight and surface area. And there's different categories under which people can fall. There was a study that was published 27 years ago now, or 26 years ago, that provided a really strong clinical study indication that being able to reduce body weight had a lot of positive outcomes. Among them was changes in susceptibility to cancer or diabetes, reduction in premature death, as well as changes across blood pressure, fasting glucose levels, as well as lipid counts. And so the objectives of our lecture now, we're going to try and tackle the first couple parts of metabolism and ATP production is what we're going to first focus on. We're going to talk about the origins of free radicals and try and look at how fats are synthesized and how they're used, as well as some of the major diets and how they kind of use this information, as well as perhaps what might interest some of you is the metabolism of alcohol and how that can have in excess deleterious effects. And so first, tackling the first phase of metabolism is going to be glycolysis that we're going to talk about today. So the fundamental energy currency of all of our cells across prokaryotes and eukaryotes is ATP, adenosine triphosphate. And so there are multiple times throughout this entire semester where we've talked about ATP in that it provides energy for the catalysis of different reactions through the activation of signaling cascades like the RAS-MAPK cascade with those kinases. ATP lies at the heart of that, as well as with motors and pumps that require active energy. ATP, again, lies at the heart of all of that. And so we talked about how ATP is hydrolyzed and how the phosphodiester bond is broken and how the gamma phosphate is released with two hydrogen molecules to produce ADP and this inorganic phosphate that's oftentimes used for different functions. It's important to note that we've talked about how ATP is used, but now we're going to focus on how ATP is produced. The synthesis of ATP is incredibly important, and it happens across different environments and different contexts. And so it's important to be able to, have the ability to synthesize ATP in different environments. And the main distinction or the main differences in those environments that have evolved over time are in the presence or absence of oxygen. The production of ATP or the synthesis of ATP in the absence of oxygen, we refer to the absence of oxygen being anaerobic, typically relies on the use of what we call fermentable substrates that produce either lactate or ethanol along with the carbon dioxide or ethanol with carbon dioxide. And so the fermentable substrates, or that fermentation, is typically what we talked about. We use that to describe the generation of ATP in the absence of oxygen. In the presence of oxygen, we usually refer to that as an aerobic process. And when we have in the presence of oxygen, that permits oxidizable substrates to undergo reactions that produce lots of ATP, as well as ending up with water and carbon dioxide. What's important to note is that while both eukaryotes and prokaryotes have pathways, use the anaerobic pathways, with oxygens, in the presence of oxygen for the aerobic pathway, eukaryotes have used that to over time, develop very efficient pathways that produce lots of ATP when there is oxygen available. And again, some tissues under certain conditions use both. An example is our heart. Whenever there's an excess of whenever there's the requirement to develop ATP, it's able to leverage through the expression of certain enzymes, the anaerobic pathway. And as well as I'm sure many of you can appreciate, and another tissue type that does that is skeletal muscle. Skeletal muscle is able to use the products of anaerobic pathways to use those substrates for producing ATP. So the oxidation of different molecules is incredibly important, and it warrants taking a moment to set the stage of oxidation so that we can better describe glycolysis. As I'm sure all of you are aware of from your chemistry classes, from your high school chemistry classes, as well as perhaps other classes you've already taken, oxidation involves the loss of electrons. And so a lot of our metabolic substrates that we're going to talk about throughout this lecture, as well as the next lecture, involve and particularly important for eukaryotes, oxidizable substrates. And the oxidation, again, the loss of electrons is highly exergonic. And so what that means is that it has, it's highly energetically favorable. It has a negative delta G, and that means that these are going to be preferred pathways when they're chemically possible or chemically available. And so as an example here, we have ethanol that when undergoing oxidation becomes acetaldehyde and produces or loses 2 protons in the form of hydrogen and 2 electrons. Typically, you can, during these oxidation reactions or reduction reactions, typically hydrogens and electrons kind of follow suit. And so when you see when you see 2 electrons being lost, you'll typically see hydrogens following suit. And so whenever there is the loss of hydrogen ions here, we typically refer to them as a dehydrogenation event. And as you can imagine, the enzymes that catalyze these reactions are called dehydrogenase. And so in this example here, ethanol undergoing this oxidation and going under dehydrogenation, the enzyme that catalyzes this is called alcohol dehydrogenase. So when these electrons are being lost, there needs to be, these electrons that are going to be lost are going to be critically important for the end product of metabolism. And they're going to be, and typically the loss of electrons carry energy with them that mediates the downstream events that are important for the generation of ATP. But it's important to have intermediaries that will carry these electrons to their final destination to mediate the generation of ATP. And so we're going to have electron acceptors that help carry the energy associated with these electrons downstream. And so they're going to eventually go to, their eventual destination is going to be on oxygen. But again, we're going to have these donors that help carry them to that destination at the end. And so these intermediate electron acceptors, or then eventually donors to oxygen, we call them coenzymes. And so if you remember, we talked about coenzymes and cofactors many lectures ago. And so what the important distinction here is that coenzymes here will typically temporarily accept to later donate the electrons for ATP generation, and that they function along with enzymes as their substrates. But again, what's important to know is that they're not consumed here. The distinction between coenzymes and cofactors, typically cofactors are ions, they're typically small molecules like ions that again are not consumed, but the distinction here is that coenzymes are typically larger molecules and cofactors are typically smaller molecules. The most common coenzyme or electron acceptor that we're going to refer to throughout this lecture and the next lecture is called nicotinamide adenine dinucleotide. That's going to be my last time saying the whole name. We usually refer to it as NAD. When NAD accepts a proton and two electrons, the proton in the form of a positive hydrogen ion, it will become its reducing equivalent. So here it's reducing, it's gaining an electron. As I'm sure many of you are, maybe you guys are aware of this. I can remember the first 2 years of BME curriculum, the way to distinguish oxidation versus reaction, I always would whisper to myself, oil rig. Oxidation, you lose; reduction, you gain an electron. Oil rig. I'm really happy to see some nodding because I was really afraid I was going to sound like really old. So typically, with the reducing equivalent of NAD that gains the electrons becomes NADH, and so it's two species. It's basically two versions of the same molecule, but what's happening here is that with NADH, that's the reducing equivalent that has gained the electron. And so when you have NAD with two hydrogens without the positive charge that has the electrons with it, you're going to produce NADH with an extra hydrogen positive ion. It's the same thing fundamentally. And so this is the way that electrons will be stored in some way of potential energy, because again, they carry energy with them that'll have downstream consequences for the production of large amounts of ATP. And so the structure of NAD is you have your adenine, your ribose sugar. You all, I'm sure, are very well acquainted with phosphate groups now. And so we have two phosphate groups here to create a pyrophosphate bridge. And so here we have nicotinamide in its oxidized form with the absence of the electron. And once it undergoes reduction into the NADH form, this is what it looks like. And so we're going to be referring back to NAD or NADH to refer to the donating or accepting of electrons throughout metabolism. So glycolysis is is going to be breaking down glucose to yield ATP. That is the fundamental breakdown of glycolysis. The input is glucose. The output, the main output, is going to be ATP. The reverse process of that, the production of glucose, is a process called gluconeogenesis, and we're going to be talking about that a couple different points. And so what's important to note is that glycolysis is referring specifically to the anaerobic portion of metabolism. This is not a portion that's dependent on the presence of oxygen here. There's going to be the role of oxygen, whether it's present or absent, is going to happen after glycolysis. And what's going to end up happening is that in the absence of oxygen, we're going to have fermentation, which is the metabolic breakdown with no net oxidation. So that's why the outcomes of anaerobic metabolism are typically referred to as fermentation. So for this class, I am not going to ask you to memorize every single piece of glycolysis. If you take a future biochemistry class, you will likely be required. If you were taking the MCATs, it would behoove you to memorize them later on when you get closer to that. But for the context of this class, I'm not going to be asking you to memorize each step, each enzyme of glycolysis, but rather appreciate glycolysis. And basically it's a story of 3 acts, for lack of a better phrase. And I'll be asking you to at least memorize, or at least know the general takeaway points that I'm going to describe for each act of glycolysis here. And so the main picture, so this is important for you to note. Again, the input for glycolysis is glucose. The output, 1 glucose, 1 6-carbon glucose. The output is going to be 2 pyruvate molecules, as depicted here, this 3-carbon ring, or this 3-carbon structure. And the way we get there is going to be broken down as preparation and cleavage, oxidation, and eventual pyruvate formation. Before we jump into glycolysis, are there any questions about oxidation, about free radicals, fermentation, anything we've covered yet so far? Any questions? Yes, Joy. Yeah, the input is 1 glucose. The output is 2 pyruvate molecules. That does not account for the ATP that is produced, but rather is focusing on the molecules that are going to be produced beyond ATP. Are there any other questions? Yes. Yeah.
So glycolysis is specifically the anaerobic portion of metabolism. So the aerobic part, is that like respiration or?
Yeah, it will be, and we're going to get to that. That's going to be the subject of the next lecture. Okay. So what I want to take a moment to say really quickly that I may not have expressed clearly in the previous slide. So the aerobic and anaerobic parts of metabolism. The aerobic parts are going to be what's going to happen after glycolysis if there is oxygen available. Glycolysis is not dependent on oxygen here. So it's an anaerobic pathway, but the main it is an anaerobic pathway. But then also after glycolysis, there's going to be the potential for an anaerobic or aerobic pathway after that. There's a slide that I have that hopefully we'll get to in time, but will help delineate that or maybe illustrate that a little bit more clearly. But the key thing that I want you to know here is that glycolysis is an anaerobic pathway, not dependent on oxygen, and then will produce 2 pyruvate molecules. What happens with pyruvate then is dependent on oxygen, but that'll be what hopefully we have time and we'll get to a couple of the enzymes that can help dictate that pathway as well. Yes. No, so fermentation, that's a good question. So after glycolysis occurs, in the absence of oxygen, one of the things that can happen is fermentation. All right, let's jump into it. So preparation and cleavage. We have one glucose molecule, and everything's going to be depicted structurally down below. And so glucose can be, we oftentimes see glucose in a 6-carbon ring structure. Typically for glycolysis, it's oftentimes going to be illustrated in a linear structure. And that is going to be a much easier way to appreciate what is happening and where things are happening on each molecule we're focusing on. And so what's going to end up happening is that it might seem backwards, but we're actually going to be using ATP during this process. And to help, one of the things that I remember a professor saying is to help understand this a little bit better is, you got to use money to make money, for lack of a better phrase. And so what's going to end up happening is during the preparation and cleavage phase, we're going to be having glucose that's going to become glucose 6-phosphate after the hydrolysis of this ATP molecule by hexokinase. And once that happens, there's going to be another event that's going to, another enzyme is going to now basically create like a mirror image of the other end of glucose 6-phosphate, which now will become fructose 6-phosphate. We call that isomerization, and if you can imagine, the enzyme that does this is isomerase. Finally, there's going to be the hydrolysis of another ATP that's going to add a phosphate group to the other end up here. And so we have 2 phosphorylation events. That's what those 2 phosphates are doing here. And now what's going to end up happening is that the end result is going to, if you can appreciate this, there's the fructose 1,6-phosphate, or fructose 1,6-biphosphate, it's also called. It's a bit of a mirror image of itself if you look at the top and bottom. And so this is going to be cleaved to produce two molecules called glyceraldehyde 3-phosphate, or G3P. That's the easier way to refer to it. And so the preparation and cleavage phase involves 2 ATPs being consumed to produce 2 G3Ps. Are there any questions on this slide? All right. Now, I'm going to try and make sure that this is clear starting from this slide forward. The last slide produced 2 G3Ps. Starting from here, we're going to be focusing on 1 G3P at a time. So basically, everything that's produced on this slide, you can imagine there's 2 of, if that helps. And I'll be trying to continue saying that to remind you all, as well as to remind myself. So what ends up happening, starting at the top with 1 G3P molecule, we're going to end up having an inorganic phosphate that's added onto our G3P, as well as the loss of an electron, oxidation event, where NAD will now take up and accept that electron. And so this oxidation event, as well as the addition of this phosphate group, produces what we call 1,3-biphosphoglycerate. And so in addition to that, we now have another, we're going to have, instead of the consumption of an ATP, we're now going to be producing an ATP. And so the input again here is going to be 1 G3P, 1 glycerol 3-phosphate, and an NAD as well as an ADP. So now again, the NAD is going to be serving as the electron acceptor, forgive me, as well as now producing 1 ATP. And so this ability of a transfer of a phosphate group from ADP to ATP by the substrate being 1,3-biphosphoglycerate, the ability now of an enzyme to act on it and to mediate the transfer of a phosphate group to ADP is called substrate level phosphorylation, where the substrate of the enzyme is mediating phosphorylation. And so now we've produced one ATP, but again, this is for each G3P. So overall, we've produced now 2 ATPs. And so what ends up happening now is that at this point, at the end of this oxidation step, we're going to have two ATPs that we've produced, but there's no net gain because we've used 2 ATPs to get to this point. So the output here is 3-phosphoglycerate, 1 NADH, and 1 ATP. The NADH will come in hand pretty soon. And so finally, what's going to end up happening here is the last step is pyruvate formation. So now we're going to be reading from right to left on the bottom for when you want to try and look back at the structures here. So we have 3-phosphoglycerate. There's going to end up being the addition of a phosphate group. And in addition to that, there's going to be a condensation reaction where water is released, creating now this on the bottom second image from the left, this enol group here. This is now phosphoenol pyruvate. And so the formation here, what's going to end up happening is there's going to be another substrate level phosphorylation event where we produce an ATP. But again, this is all, we're still talking about at one G3P molecule. So once that substrate level phosphorylation occurs, the loss of that phosphate group or the donation of it is going to produce pyruvate here. So with each 3-phosphoglycerate, we produce 1 pyruvate and 1 ATP. All right. And so this also will happen twice for each glucose molecule that we consume. And so the net output here from glycolysis so far is for each glucose, we are going to be netting 2 ATPs, 2 pyruvates, and 2 NADHs. This is important to know. Virtually every single cell relies on this pathway. Glycolysis is incredibly important for prokaryotes and eukaryotes. And so what will happen after this, and this goes back to a question a moment ago, what happens after this will now depend on the presence or absence of oxygen, whether it'll be anaerobic or aerobic pathways that emerge from here. So with from here, in the presence of oxygen, so that's going to be the top right here, from each pyruvate that has been produced from glucose, what will end up happening is that there's going to be an enzyme that's going to be linked to pyruvate. It's called coenzyme A, that's going to produce an oxygen, or sorry, not an oxygen, a carbon dioxide, forgive me. And in addition to that, there's going to be another enzyme called pyruvate dehydrogenase that'll produce another NADH, and both those together will produce what's referred to as acetyl-CoA or acetyl-coenzyme A. And so with both pyruvate dehydrogenase and now coenzyme A, you'll produce from pyruvate acetyl-CoA, which is incredibly important for aerobic respiration, which will be what's discussed next class. In the absence of oxygen, fermentation takes place. And this goes back to another question we had earlier in the lecture. And so one of the NADHs that you had is going to be used here. So for the bottom pathway that usually most yeast and some other microbes will produce, they possess an enzyme called pyruvate decarboxylase. As you can imagine, a decarboxylase is the loss of carbon dioxide. And so they'll produce acetaldehyde, alcohol dehydrogenase, as well as the now reduction of NADH to give up the electrons will now produce ethanol. And so this is alcohol fermentation here. And so for those of you who enjoy making your own beer or consuming beer or other forms of alcohol, this is the pathway by which ethanol is produced. We, as eukaryotes, do not possess pyruvate decarboxylase. So the fermentation process that our cells use is lactate fermentation. And whenever you hear people talk about, or whenever you've heard referenced the buildup of lactic acid, this is what's happening. In the absence of oxygen or sufficient oxygen, pyruvate via lactate dehydrogenase and the reduction or the giving up of the electrons will now produce lactate or lactic acid in its non-protonated form. And so again, for some prokaryotes, they will go through alcohol fermentation. That's what they'll do with pyruvate in the absence of oxygen. For eukaryotes, we'll go through lactate fermentation. The appropriate question is, what is done or how is lactate used? I just went through that entire process without these slides, sorry. Lactate is going to be used in what's referred to as the Quarry Cycle here. Typically, to simplify the, excuse me. Typically in the Quarry Cycle, excuse me, I'm going to take a moment to drink some water. Are there any questions? So typically with the buildup of lactate, as an example, you can think of the buildup of lactate skeletal muscle during heavy exertion. For example, someone who's a sprinter, that's going to be a great example of an anaerobic event. The buildup of lactate is going to go through the process of gluconeogenesis. We talked about how that's the kind of reverse pathway of producing glucose instead of breaking it down. And then the name also kind of helps to provide the definition. Gluco for glucose, new, genesis, formation, so the new formation of glucose here. And so in the liver, the lactate is going to be transported out to the liver. And what's going to end up happening is that it's going to possess enzymes that's going to produce 2 pyruvates from lactate. And within the liver, with the consumption of NADH, you're going to produce glucose that's going to go back to the skeletal muscle as an energy source. And so the main take home here is that whenever we have lactate fermentation, lactate is used by the Quarry Cycle via gluconeogenesis to produce more glucose that'll then via glycolysis provide more ATP for energy. And so the similarities, the glycolysis and oh yes, Liam, do you have a question?
Is there like gene expression that will inhibit the gluconeogenesis if there's like too much glucose in blood?
Yeah, so there, hold that thought. So there's a way in which typically excess glucose is stored. So hold that thought. There's for genetic regulation, that's a good question. I don't, there are certainly some enzymes that that in different populations that have different expressions of different of some of these enzymes. And so you can imagine, so we haven't gotten to aerobic respiration yet, but my family comes from Ethiopia, and my dad's family comes from the region where a lot of the distance runners come from. And so it's believed to be said that some of the runners have, some of the individuals from that region of Ethiopia have different expression levels of enzymes that help facilitate more efficient respiration. We haven't gotten to that part, but like that's like one example. This is somewhat related to metabolism. There are certain populations that have different levels of alcohol dehydrogenase that makes metabolism of alcohol difficult or less efficient. And so there's definitely a genetic difference or genetic regulation that might exist within some populations that might alter some of these pathways. But to the storing of excess glucose, we're going to get to that in a moment. Yes, Edward. That's a great question. So if you remember, what's happening here is glycolysis produces 2 pyruvates. In an anaerobic environment, in eukaryotes, each pyruvate becomes a lactate molecule. And so now through the Quarry cycle, it's going to go back and become glucose, which now can be used to produce, it's not as efficient as alternatives, but we'll be able to produce 2 ATP for each glucose that's going to be metabolized via glycolysis. Then we'll get 2 pyruvates again. We're still in an anaerobic environment. That will become lactate. It'll be sent to the liver to produce glucose again. And so it's less efficient than what we'll talk about next class. But with each glycolysis event, we're going to be producing 2 ATP. And as you can imagine, in a physical exertion event that is anaerobic, like sprinting for 100 meters, or someone who's training to be a sprinter, these events, these Quarry cycle processes are happening incredibly quickly because there's huge demand for ATP, and so it's not just a loop. As you can see here, this is producing 2 ATP each time glycolysis is occurring. There's also a cost to making it, but again, this is why it's far less efficient than the alternative of aerobic respiration that eukaryotes possess. Not the liver, but rather access to oxygen through the bloodstream. Does that make sense? Yes. Does that make sense? So what I should say is it's not binary, if that makes sense. It's not all one or all the other, but rather when we think about the absence or presence of oxygen, we usually refer to that as oxygen tension. And so it's typically a balance of, if there's not sufficient enough oxygen in this moment, we'll go through this pathway. If there is, we'll go through aerobic respiration. But it's not, I want to make sure I say this clearly, like it's not a simple binary that it's only one or the other. They're not mutually exclusive, but rather it's typically a balance based on the current snapshot in time of what the oxygen tension is like, the balance of oxygen. Does that help make sense? Because it's not just going to be all the cells, not all of the cells in my quadricep or my hamstring during a sprinting event are going to be all going through lactate fermentation. Does that help? Yes. Yes, yeah, and so the presence of ATP. There will be ATP available in the liver by cells that are producing it. So it's not as if ATP is not available for this process to occur, but it's helping to meet the need in the moment. Does that make sense? Exactly. Yeah. I'll go ahead and put a pin in it there, and we're going to have a guest lecture for next class, but I'll try and provide a small addition of this on the recording for your notes for next time.